All right, so welcome back to this Go with the Flow series. Today we are going to do an exciting start to a different part of the series. We're done with our 7 introductory videos, and after that we enter a new phase. And for this new phase we have a special guest on today, which is Franklin or Buddy Fuda from UT Southwestern in Dallas. So Buddy and I spent lots of years at UT Southwestern. He was actually a resident, then fellow, and now he's the director of the flow cytometry lab there, knows all the intricacies of cluster analysis and high-end flow cytometry. And he has agreed to join in this new series that we have decided to call, well, first, turns out Buddy has a new computer, and so it doesn't have a camera yet, and so you cannot see him. So he agreed for me to use this picture from their website. So that's Buddy. And this new series we are going to call Free Flowing. And what that means is we may discuss thematic topics, we might do random cases, we might have different guests coming in and doing this. But for the very first one, what we are going to do is focus on CD5 positive B-lineage lymphoma. So Buddy is going to take us through that journey. And the reason we are calling it a video atlas actually has history. Both Buddy and I have spent a lot of conversations about eventually doing a flow cytometry atlas. Buddy has been stashing away interesting cases for it. But we have realized that it's probably not going to happen. And so what we decided to do is might as well do a video atlas where you will get to hear us go over nuances, which is much better and far more informative than a dry reading of a book. So, and it's going to be free. That's why free flowing. So today, our focus is CD5 positive B-lineage lymphomas. And you remember this from the introductory series. If you've watched it, there's a B-cell algorithm that takes you through CD5, CD10. And what we're going to focus on is this end of the algorithm, CD5 positive B-cell lymphomas. So with that, I'm going to stop sharing from this end in Iowa and let Buddy share his screen. Buddy, are you able to do that?
Okay. Is it, are you seeing it?
Yeah, you might want to put it into the slideshow mode.
There we go.
Perfect. Yep, go for it.
Okay. All right. Well, first I want to thank Nitin for inviting me to partake in this series. Very excited about it. Like Nitin had mentioned, we had spent many years talking about doing an atlas, but that can be time consuming and very difficult. This seems like a really nice forum or platform to share these interesting cases. I've spent the past 6 or so years trying to get Nitin to come back to Texas because we really miss him here. But I guess a virtual Nitin is better than no Nitin at all. So really enjoy the opportunity to work with you again, Nitin, on a project. So yeah, I will go over some CD5 positive B-cell lymphomas. And this is not going to include all the CD5 positive possibilities for B-cell lymphoma, but it will include the most common and most characteristic ones. So when we talk about flow cytometry, I always teach my residents the first place that I like them to start is to look at the physical parameters of the cell populations because it gives us a good starting point for developing a differential diagnosis. So I just wanted to go over the physical parameters real quickly just to remind everybody what exactly you're looking at here and what the physical parameters can actually tell you. So here we've got an actual cytospin, and the cytospins correspond to the plots above them. So these are actual cases. Here in the cytospin, you can see that if the red blood cell here is a reference, you can see we've got a lot of small lymphoid cells sitting here through here. So when we look at the actual flow plot, these lymphoid cells are colored red up here. And we can see that they sit kind of over here toward the origin on this forward light scatter axis. So forward light scatter is based predominantly by cell size. The larger a cell is, the further along the axis the cell will travel. So here, these are small cells on the cytospin, and you can see that they do not travel very far along the forward light scatter axis either. If we move over here to the second case, again, using our mature red blood cells as a reference, these cells here are larger than these cells that we saw over here. They're kind of medium sized. So if we look up the flow cytometry plot, we can see then that they moved further along the forward light scatter axis. So they have higher forward light scatter, or what is most people call moderate forward light scatter, and that correlates to medium cell size. These cells over here are pretty much huge. So these are very large lymphoid cells, and we would expect them to have very high forward light scatter. And if you look over here on the forward light scatter axis, the red population represents these cells, and they have very high forward light scatter. So that's how, using forward light scatter, we can get an idea about what kind of cell population we might be dealing with based on the cell size. Now, orthogonal light scatter, otherwise known as side light scatter, we plot here on the y-axis, is going to be determined predominantly by what we call cytoplasmic complexity. And in general, that means how granular the cytoplasm is, or how many vacuoles are in it, things like that. So you can see this population that's way up here. These are this gray population up here. These are granulocytes. Now, our small lymphocytes sitting here don't have any granules or vacuoles in them at all. So you can see that they kind of sit down here near the origin for the side scatter axis. Okay, so that makes sense. Same thing with this, this plot over here. These medium-sized lymphoid cells, they really don't have much in the way of granules or vacuoles, so they're very low side scatter. Whereas these very large cells over here have very prominent vacuoles, and that's why we see on this plot here that these cells actually do show higher side scatter than your small lymphocytes painted green here. Okay, so again, we always start with the physical parameters just to give us an idea of what size cell population or what size the cells in the population should generally be, and it helps us with our differential diagnosis and our approach from that point. Now we're talking about B-cell lymphomas, so I just wanted to show a slide with the basic B-cell markers that are looked at in flow cytometry. CD19, CD20, and CD79A and B, and immunoglobulin light chain, as well as FMC7. These are all B-cell specific type markers. These other markers here, CD22, CD23, CD24, are not necessarily specific for B cells, but they're B-cell associated markers. And so these are the markers that we look at routinely when we're looking at B cells in flow cytometry. One of the most important or useful markers that we have in assessing B lineage cells by flow cytometry is surface immunoglobulin light chain or cytoplasmic immunoglobulin light chain. So what we do is we typically plot lambda versus kappa. And then when you do that, unlike a quadrant analysis, we have 1, 2, 3, 4 quadrants, you kind of look at these and you say, I've got a diagonal, and anything along the diagonal I'm going to consider to be negative for the immunoglobulin light chain. And the positive thing kind of fall off of the diagonal into these regions over here. So you can see here, this red population is negative for surface immunoglobulin, whereas these two blue populations here and here are positive for surface immunoglobulin. This population's lambda, this population's kappa. Now, if we look at these representative plots down here, we can see we want to focus on the red population here. This would be considered negative for surface immunoglobulin because it's sitting along the diagonal. Here's a cytoplasmic tube with kappa versus lambda. This red population here is also considered negative because it's sitting along the diagonal. And then if we look over here at this population, right here, we can see that if this is our diagonal, this population falls off of the diagonal toward kappa. And so this would be a monotypic population of B lineage cells here. And the reason immunoglobulin light chain is so useful in flow cytometry is because most B cell neoplasms that are mature will be monotypic. They'll show restriction to either kappa or lambda. Now that's not always going to be the case, and you might have a population like this over here where it's a B-cell lymphoma that doesn't express either kappa or lambda, but that is still considered an abnormality and suggestive that of a B-cell neoplasm. One important note to take into consideration when we're talking about immunoglobulin expression and looking at it by flow cytometry is that if a population lacks expression, a B-cell population that is lacks expression of immunoglobulin light chain, you might, if you have it, you can change the actual preparation of your antibody. And so here you can see that this is a monoclonal preparation of kappa, a monoclonal preparation of lambda, and these cells are negative for surface immunoglobulin when looking at this plot. This is the exact same case, and we can see that this population actually does express dim lambda. So it kind of falls off of the axis here. And this is a polyclonal preparation. So the polyclonal preparations are going to look at multiple epitopes on your actual molecules. And so for whatever reason, the epitope wasn't being expressed over here, but they actually did truly have lambda. So the polyclonal population showed that. So we would look at this and say, yes, we've got a lambda restricted population of B cells. Now, that doesn't mean that if we didn't have the polyclonal, we would miss this, because if it's a mature B cell population and it lacks expression of surface immunoglobulin, we would also consider that abnormal. Okay, so this is our algorithm that Nitin had showed us earlier, and we're going to focus again on the CD5 positive lymphomas and, for the most part, the small cell lymphomas. So when we talk about CD5, I'll go back here for a second. When we talk about the CD5 positive small cell lymphomas, the two lymphomas that are most characteristically going to show that immunophenotype is chronic lymphocytic leukemia / small lymphocytic lymphoma versus mantle cell lymphoma. So those are the two we're going to focus on first. You can see here that Nitin has listed lymphoplasmacytic lymphoma in this category as well. Here it sits in larger letters over here. The reason it's sitting in larger letters over here is most commonly it is 5 negative, 10 negative, but sometimes it shows up as 5 positive. Usually when it shows up as 5 positive, it's going to be dim positive for 5 or weak positive for 5 or partial positive. Okay, so let's focus again on mantle cell lymphoma. I don't know. Okay. Mantle cell lymphoma versus CLL. When we've determined that we have a monotypic population of CD5 positive small B lineage cells, our next step is to try to figure out if it's most characteristic of one of these two types of lymphomas. And so the markers that are most commonly used are going to be FMC7, CD23, and or CD200. So we like to use, if we're going to use CD23, we like to use it in combination with FMC7. And this is what we used to do historically in our lab. We added on CD200 to that, and we were looking at all 3 markers for a while, and then we went ahead and got rid of FMC7 and 23, and now just use CD200 in our laboratory. So if you see here, you can see that mantle cell lymphoma is going to characteristically show positivity for FMC7 and negativity for CD23. Whereas CLL, SLL will be positive for CD23, but negative for FMC7. Now, FMC7 is actually an epitope of CD20, all right, but it's only expressed at certain stages of reactivity on normal B cells. So every CD20 positive B cell doesn't necessarily have to express FMC7. So it's a B cell marker. So when we in the context of CLL versus mantle cell lymphoma, one of the things that I routinely tell my residents to try to remember which is which as far as this staining profile goes. If you remember that FMC could stand for mantle cell lymphoma, it makes it easy to remember what the profile of FMC7 versus CD23 are. All right, the point I was making before, though, is that since this is actually just CD20, it makes sense that it's not specific for mantle cell lymphoma. And by no means is it specific for mantle cell lymphoma. But in the context of CLL, SLO versus mantle cell lymphoma, you can kind of use that to remember how that staining profile works. Okay, so then CD200, CD200 is a marker that's going to be expressed by CLL. So you can see it's positive for CLL here, but negative for mantle cell lymphoma. Again, CD200 is not a specific marker for CLL. In fact, most B-cell lymphomas do express CD200. It's just that mantle cell lymphoma is typically negative for CD200. So we can use that to help differentiate the two. All right, beyond that, once we've assessed these markers and we have a pretty good idea that we're dealing with either CLL or a mantle cell, we can look at some of the other markers for characteristic features of those two lymphomas. So one of the markers that we look at, and we call these supporting features, is CD20. And you can see here, painted in blue, these are normal polytypic B cells. Okay, so for CD20, you can see the polytypic B cells sit right here. Now we always, when we call something dim positive, positive, or bright positive, it's based upon the fluorescent intensity of the normal polytypic B cells. So if you look here, our lymphoma population is CD20 bright positive. So compared to the B cells here, if you took the median of the polytypic B cells here and looked at the median of these monotypic CD5 positive B cells, these would be bright positive for CD20. Now that's kind of what mantle cell lymphoma typically shows. It either shows bright positivity or the same intensity as your polytypic B cells. Chronic lymphocytic leukemia, small lymphocytic lymphoma, on the other hand, is characteristically dim positive for CD20, partial dim positive for CD20, like in this case, or completely negative for CD20. So the blue cells here are normal polytypic B cells. You can see the nice normal fluorescence intensity here, and the red population is much dimmer to that, to negative. All right. Then we look at our surface immunoglobulin light chains. If you go back to mantle cell lymphoma, again, the diagonal is negative, and then our positive populations fall off the diagonal, and here's our normal polytypic B cells painted in blue. Our red population represents our CD5 positive B cell lymphoma, they're monotypic B cells. And here we have nice bright intensity of kappa. So you can see if you look along this axis, the median of the red goes beyond the median of the blue here. So that's nice and bright. So again, that is a nice characteristic feature of mantle cell lymphoma, to show either bright expression of surface immunoglobulin or the same intensity as the polytypic B cells. Whereas the chronic lymphocytic leukemia, small lymphocytic lymphoma, typically shows dim to negative expression of surface immunoglobulin light chain compared to your polytypic B cells. Okay, so we can use those features as supportive evidence that we are making the right differential diagnosis based off of our findings on FMC7, 23, or 200. Now, I just want to mention one caveat when we're talking about CD200. So because we're using CD200 to try to differentiate CLL versus mantle cell, it's very important to recognize that there is a subset of non-nodal mantle cell lymphoma, and these happen to have an indolent course, that actually express CD200. So it's really important, again, to look at the case all together and put all of your features together when you're coming up with your differential diagnosis. So in those cases, your surface immunoglobulin light chain and your CD20 would likely be bright positive, but then you would see a CD5 positive lymphoma that was CD200 positive. And so then you would do the molecular type studies and genetic studies to see if you're dealing with one of these subsets of mantle cell lymphoma. I put down here the reference for the paper that actually talks about that. Okay, so we're going to go ahead and show an example approach of what we do when we're assessing a tissue and we discover that it's got a B-cell lymphoma in it. So here we've got the blue are going to be reference mature polytypic B lymphocytes. The green are going to represent reference T lymphocytes. And the red are going to be our cells of interest. So they're going to be our abnormal cells. So we go through here, and the first thing, again, that I look at is that the forward light scatter on the red sits at the same level of movement that the mature B cells and the mature T cells sit. So these should be small cells then, my red. Okay, they have very low cytoplasmic complexity, so it makes sense that they probably don't have a lot in the way of granules or vacuoles. Now, they happen to be CD19 positive, they're CD20 positive, and they have surface immunoglobulin light chain, so this tells me that these should be B lineage cells. Since they only express kappa, so they fall off the axis towards kappa, and they don't have any lambda, these are monotypic B cells. So now I know I've got monotypic B cells. That tells me I've got an abnormal population of B cells. Now, abnormal doesn't mean malignant. It just tells me that I've got a monotypic population. So monotypic most likely means monoclonal. Okay, so most of the time when you have a monotypic B cell population, it's going to be rising from a single cell, and it's monoclonal. That doesn't hold up 100% of the time, though. Monoclonal populations are most likely going to be neoplastic and most likely going to be a malignant neoplasm. Okay, but that doesn't hold up 100% of the time either. But that's what is most characteristic. So at this point, we would say that we've got here a population of monotypic small B lineage cells. All right, the fact that they have surface immunoglobulin light chain then tells me that these are likely to be mature, okay? So most characteristically, if you have surface immunoglobulin light chain expression on an abnormal B cell population, you're dealing with a mature B cell population. So here then, I would put that all together and I'd say, well, I think what we're dealing with is a mature B lineage non-Hodgkin lymphoma of small cell size. Okay, now once I have that, I then go back to my algorithm. And I'm going to start my algorithm with CD5 and CD10. So here I can see that that population is nicely CD5 positive, it's CD10 negative. So that gives me a differential then based on my algorithm of CLL, SLL versus mantle cell lymphoma. Then I'll go ahead and I'll look at my other markers that I mentioned earlier that can help differentiate between the two. In this case, our CD23 is negative. Our FMC7 is positive. And again, looking at that pattern, I can say, okay, when I'm looking at this and I'm in the context of CLL versus mantle cell, the FMC7 can mean for mantle cell lymphoma. So this ought to be a mantle cell lymphoma. So more than likely, we're dealing with a mantle cell lymphoma in this case. Now, the FMC7, actually, I can't remember what the F stands for, but it's some kind of medical center in either Australia or New Zealand or something like that. All right, so based off of those findings, this is an example of the interpretation that I would release. So in my interpretation, I'm going to go ahead and I'm going to give them my findings. So I'm going to tell them the findings show a monotypic population of small CD5 positive B lineage cells, and this suggests a B lineage non-Hodgkin lymphoma of small cell size. All right, after I do that, I'm going to tell them what it is most characteristic of. So based off of my immunophenotype, I'm going to make a differential diagnosis. So here, the immunophenotypic differential diagnosis is going to include, but it's not only going to be limited to mantle cell lymphoma. So based off the immunophenotype, that's what I would expect this to be, but it doesn't necessarily have to be that. Next thing I would do is give them any kind of further studies that might be helpful. So here I would say if clinically warranted, further evaluation could include fluorescence in situ hybridization for a T1114 translocation and the gene for cyclin D1, which is the gene that is translocated in mantle cell lymphoma, sits on 11. And then on 14, you've got the immunoglobulin heavy chain, so you get a translocation between these two. Or immunohistochemical staining for cyclin D1. Because in mantle cell lymphoma, you get overexpression of cyclin D1. Now, next in my interpretation, I'm going to remind everybody that the WHO requires more than just an immunophenotype to make a diagnosis. So I'm going to tell them that final interpretation is going to require you to use the immunophenotype in correlation with the clinical features, the morphologic features, and the genetic features. All right. And then lastly, I'm once more going to highlight that flow cytometry itself is an ancillary test and it is not a final diagnosis. The reason that we go through highlighting that these things are not diagnostic is because sometimes people will look at this and they'll see your differential diagnosis, or they'll see something saying most consistent with or characteristic of, and they may think that that is the bottom line diagnosis. And we know that flow cytometry gives us an immunophenotype, which is only one piece of the puzzle. So we need all those other things to come up with a final diagnosis. Okay, so just looking at CLL, SLL, here's our typical features. This one here happens to be small to medium sized. So you might have some pro-lymphocytes in this CLL. Again, it's going to be 5 positive, 10 negative. It's going to have CD23 and lack CD or FMC7. It's going to be restricted, show dim expression of immunoglobulin light chain, and dim to negative CD20. Very characteristic immunophenotype of CLL. Sometimes you're going to get a CD5 lymphoma that has an indeterminate immunophenotype based on those markers. So in this case, again, we've got a small cell population that shows low orthogonal light scatter. They are B lineage based on CD19, CD20 expression, immunoglobulin expression, and CD23 expression, and FMC7 expression. They're monotypic, so they should be neoplastic, and they should be mature, okay? So we're probably dealing with a B lineage non-Hodgkin lymphoma, small cell size, a mature one. They're 5 positive, 10 negative, so it gives us that differential CLL versus mantle cell. But then when we look at our CD23 versus FMC7, they happen to be both 23 positive and FMC7 positive. Now this FMC7, you might look at it and go, is it really positive? This particular one is in FITC, which is a very dim fluorochrome. So any movement along this axis is usually positive. So here we've got positive for FMC7, positive for 23. That gives us an indeterminate immunophenotype. We can then look at our supporting features to see if that helps a little bit. This particular case is bright for CD20, which is more characteristic of mantle cell lymphoma, but then it's dim for kappa, which is more characteristic of CLL, SLL. So this one truly is an indeterminate immunophenotype. We're gonna obviously need genetic testing to help us figure out what the bottom line diagnosis is. Our differential diagnosis is going to really broaden up. It could be CLL, could be mantle cell. It might be lymphoplasmacytic lymphoma, or it might be some other B-cell lymphoma with an uncharacteristic immunophenotype. Okay, this here was a case of an aggressive variant of mantle cell lymphoma. So what do I mean by that? Well, here we can see that the immunophenotypic features are typical for mantle cell lymphoma. They're showing B lineage expression. They're monotypic. They're 5 positive and 10 negative, and then they have the characteristic pattern of staining for FMC7 and 23. Here the FMC7's positive and the CD23 is negative. Now it looks like it's coming up here a little bit, but that's due to some autofluorescence. All right, so when you have an isotypic control tube or an autofluorescence control tube, you can see that this is truly negative. So here, this is very characteristic overall immunophenotype. For mantle cell lymphoma, it's got the bright kappa, it's got the bright CD20, all right? But if you notice on the forward light scatter, these cells are actually medium to large, all right? So this kind of gives you a clue when you're looking at flow cytometry alone. If you don't have the morphology, you might be clued off that potentially this could be an aggressive variant of mantle cell lymphoma. So what are those aggressive variants? A blastoid variant and pleomorphic variant. All right, this one here turned out to be a blastoid variant of mantle cell lymphoma. It did have an 11-14 translocation. Okay, so we're just going to go over some examples now. So here's some plots here, and the green is going to represent T lymphocytes. The blue is going to represent polytypic B cells, and the red is our population of interest. So again, we're going to start off on the physical parameters. So I'm going to look at my forward scatter, and I see that my red population sits at the same level of forward scatter as my polytypic B cells and T cells. So these must be small cells. They have very low cytoplasmic complexity, so they probably don't have granules or vacuoles in them. They're nicely CD45 positive. Now, CD45 is leukocyte common antigen. We use that to show that a population is hematolymphoid. If a population is positive for 45, we know that it's hematolymphoid. Hematolymphoid populations can be negative for CD45, though. All right, so we know it's hematolymphoid. We know that it's 19 and 20 positive, so it should be B lineage. It's capo restricted, so we should be dealing with a B lineage neoplasm. If we look then on our algorithm, we're gonna move to CD10 and CD5. These happen to be 5 positive and 10 negative. So here's our algorithm. Again, it places us in the CLL versus mantle cell category. Here's the CD200 on this population. It's nicely positive. So this should be a CLL SLL. We can look at our supporting features here. Some of the supporting features I didn't mention earlier, but most CLL SLLs are also DIM positive for CD45. So if your reference population is this blue polytypic B cell population, you can see that the CLL SLLs are oftentimes slightly dimmer in their intensity of expression of CD45 versus the polytypic B cells. Over here, we have nice dim expression of CD20, very characteristic of CLL, SLL versus the polytypic B cells painted blue here. So here's our CD20 coming across. And then again, we've got dim expression of surface immunoglobulin compared to these kappa expressing polytypic B cells. So CLL, SLL is what this one most likely represents. Now, when we talk about CLL, SLL, there's a few things beyond just coming up with the most characteristic features immunophenotypically that we can look for. And one of them is prognostic indicators. Now, I've listed 2 prognostic indicators that are looked at by flow cytometry currently. There's a third one, ZAP70, but I don't know too many people that are doing ZAP70 anymore. It was very difficult to do technically. So most places have moved to CD38 and CD49D. So if you look over here, this is a gated population of CLL cells. And we're going to pay attention to the CD38 here on the y-axis. So if our, based on an isotypic control tube, we see that the threshold for positivity would sit right here. So based off of that, everything above here is positive. Now, when you're talking about the prognostic indication, if your neoplastic population, if greater than or equal to 30% of the neoplastic cells express CD38, then it's associated with an overall less favorable prognosis. Okay, so you can see in this case here, we have some positive cells for CD38, but it's only 17%. And so this would be considered for prognostic purposes, negative expression of CD38. Okay, it's kind of the same thing with CD49D. When you look over here, now this population is not gated. All right, so we've got, this is an ungated plot, so you've got all of your other cells that are in the sample in here. But we can see that if this is our threshold, the bulk of the lymphoma cells express CD49D. So if we did actually have these things gated, we'd see that there'd be greater than or equal to 30%. So that's also the threshold that's typically used for CD49D expression. And it's the same thing. If it's positive, it correlates with a poorer overall survival. Now, a recent study has shown that if you've got a CLL population that has a subset that's negative and a subset that's positive, even if the positive subset doesn't reach 30%, the prognosis is likely to be the same as if it was partial positive and greater than 30%. So it probably indicates a poorer overall survival. All right, as far as CLL goes, more recently, people have become interested in looking at very high sensitivity, minimal residual disease in CLL. So they're doing this in Europe, and there's a couple institutions here in the United States that offer this test. We have just validated it, so we'll be offering it here shortly. But the sensitivity is down to 10 to the negative 5 or 0.01%. All right, so it's something that our clinicians wanted, so we brought it on board. So when you're doing this high sensitivity MRD, it can be helpful to look at additional markers for CLL beyond CD19, 20, 5, kappa lambda, FMC7, 23, and 200. So some of the markers that are looked at are CD22. And you can see here the CLL is colored in red. The polytypic B cells are colored in blue. And CLL is typically dim to negative for CD22. So here you can see along the y-axis here, it's dimmer than your polytypic B cells. CD79B, again, is another B cell marker, and CLL tends to be dim for CD79B compared to the normal polytypic B cells. Two other markers that are used are CD81. Once again, CLL-SLL tends to show dim expression of CD81 compared to your polytypic normal B cells. And then CD43 is the last marker that's typically used. And you can see here that your normal B cells are for the most part negative for CD43, but CLL-SLL is often positive for CD43. So that can be very helpful. If you get a baseline, it's very similar to doing minimal residual disease analysis for B-lymphoblastic leukemia. You want to get a baseline if you're going to run these MRD high sensitivity studies. You want to get a baseline immunophenotype. So we would basically look at these markers at diagnosis, and then on follow-ups when we're looking for minimal residual disease, we would know what they did for these particular markers to help us identify the remaining CLL-SLL. Okay. Just to carry on with CLL-SLL, you can, this isn't real common, but we see a handful of these every year, what is considered a biclonal CLL-SLL. So this here is a good example. You can see that the cells in green are your reference T cells. The cells in red are population of interest. They go from small to medium size to have low cytoplasmic complexity. They are hematolymphoid. They've got slightly dimmer expression of CD45 compared to the T cells. We can see that they're B-lineage cells because they're 19 positive. They've got CD23 and they have surface immunoglobulin. Okay, and then we get down here, again, we use surface immunoglobulin to identify abnormal populations of B cells. And if you look at this population, it looks like a polytypic population of B cells. Now, I know these are neoplastic because, you know, I did the entire analysis, but we don't have any normal B cells in this case as a reference population. So you can see there are no normal CD20 positive B cells here. Now, if they were in here, just look at this, I wouldn't know because we don't have a reference polytypic B cell population, whether this is dim expression or normal expression of kappa. But I would suspect, since I now know that this is a biclonal CLL, that my normal polytypic B cells would have been, you know, brighter for kappa and lambda. But you can see they have the characteristic features otherwise of CLL. They're CD5 positive and 10 negative. They've got 23, but no FMC7. They happen to express CD200. And so, you know, looking at the immunophenotype, we couldn't necessarily say exactly what this was, but we raised suspicion that this might actually be two separate clones of a monotypic or monoclonal B cell, or they could be a single clone that has immunoglobulin light chain switch, or they may have started the life off and then, you know, went off in both directions, lambda or kappa. All right, here's another nice example of a biclonal CLL. This time, the polytypic B cells are painted in black, and our monotypic populations are painted in red or blue. And you can see the overall features are, again, characteristic of chronic lymphocytic leukemia, small lymphocytic lymphoma. The red population is nicely CD5 positive. The blue population is partial positive. And you can see they have opposite immunoglobulin light chain expression. So this one was easy to pull out because you can pull out this population and it's going to be all lambda. And then you pull out this population here, and some of it's going to show up lambda, some of it's going to show up kappa. But then as you go through the case, you can actually see some differences in the intensity of expression of this little subset that's lambda and this red subset that's kappa. So, you know, whether or not you want to call this biclonal or two actual subsets, you can actually look at it and see that it's breaking off into one, potentially two, or potentially even 1, 2, 3. Okay. So anyhow, that turned out to be chronic lymphocytic leukemia, small lymphocytic lymphoma. This is real important to recognize that you can get these biclonal types of CLL, SLL, so that you don't miss them and just think that they're reactive polyclonal B cells. Okay, so we're going to look at another case example. Here, the polytypic B cells are painted blue, and the population of interest is red. Again, our forward light scatter shows us these are small to medium-sized cells this time. So you can see that you got small cells sitting behind the polytypic B cells, but then you've got these moderate, or these cells with moderate forward light scatter that should be medium-sized cells. They have low cytoplasmic complexity again. They're nicely CD45 positive, so we know they're hematolymphoid. They express CD19, 22, and 20, so we know they're B lineage. They're kappa restricted, so we suspect that this is a B lineage neoplasm. We're then going to move on our algorithm to the 5 and 10. They're 10 negative, 5 positive. That puts us in that same differential diagnosis, the CLL versus mantle cell lymphoma. Looking at our markers to help us differentiate the two. These guys are 23 negative, FMC7 positive, and 200 negative. So this is likely to be a mantle cell lymphoma. The supporting evidence is here that we've got nice bright expression of CD45, bright expression of CD20, bright expression of CD22, and bright expression of surface immunoglobulin light chain. Again, none of these things are specific, but when your differential diagnosis is CLL versus mantle cell lymphoma, this bright expression of these normal B cell markers is characteristic of mantle cell and not characteristic of CLL. So overall, we figured this was going to turn out to be a mantle cell lymphoma. And lo and behold, it did have a FISH positivity for translocation between 11 and 14 chromosomes, and it was cyclin D1 positive for immunohistochemical staining on the morphologic preparation. Okay, so we'll move to another example here. This time, the green is going to represent normal T cells. The B, again, normal polytypic B cells. Our population of interest is red. Our forward light scatter shows that these are small cells. They're sitting with the other small lymphocytes. They do not have a lot of cytoplasmic complexity, so they probably don't have a lot of granules or vacuoles. They are nicely CD45 positive, so they are hematolymphoid. They express both CD19 and CD20, so they're likely to be B lineage. Here we can see that they do express surface immunoglobulin light chain and that they're restricted. And on the monoclonal preparation, they have nice dim expression of lambda. On the polyclonal preparation, they either show dim to negative expression of surface immunoglobulin, and it would be negative for lambda. Moving back to our algorithm, our CD10 is negative, our CD5 is negative. Okay, so if we look at our algorithm, that puts us into a marginal zone lymphoplasmacytic or hairy cell leukemia, right? All right, so I bet you weren't expecting that when we were looking at the other markers. So let's just go back and look at these again real quick. We see that our CD20 is nicely dim positive. Our lambda is dim positive. Our CD22 is dim positive. Our CD45 is dim positive. We have CD23, but no FMC7. That sure does sound like CLL, SLL, doesn't it? Now, again, these features are not specific for anything, but they're very characteristic of CLL, SLL. It just so happens that this case does not have CD5. This actually did turn out to be a CD5 negative CLL, SLL. So you don't want to, be overly rigid in using CD5 and CD10 to put you into those diagnostic categories. When everything else looks like something, you're going to want to go, okay, well, these features actually can be seen in CLL, SLL, and potentially this is a CD5 negative variant of that. So this is a really nice case. We probably get, maybe 10 to 15 CD5 negative CLL, SLLs every year. Obviously, we're not making that diagnosis on the immunophenotype alone. So again, it's really important to recognize that the immunophenotype is only a single piece of the puzzle. Final diagnosis requires everything else. Okay, so let's move on to another. This is our last example. Here are, I'm going to go through here. The green are going to be T cells. The red are a population of interest. And the blue, I'm not quite sure what they are yet. They're probably a population of interest too, since I've mentioned them and put them on here. All right, so CD45s are leukocyte common antigen. All three populations are positive. Our red is nicely positive. Our blue is positive as well. So these are all hematolymphoid cells. If we look over here, both our red and our blue are 19 positive. So they're probably B lineage. Our CD20, our red is nicely bright positive. Our blue is actually partial positive. Okay, so again, these are probably B lineage cells. Now, I want to point this out to you guys because I didn't do this purposely, but it's kind of a nice representation of this. You can look at flow cytometry data using either logarithmic scale or a bi-exponential scale. When we were doing 4-color flow cytometry, we always looked at logarithmic scale. Now that we were on the 10-color machines, we're typically looking at bi-exponential scales. So what the bi-exponential scale does, see these cells here, these look nicely positive, positive, positive. But see this line of cells sitting down here at the origin for this y-axis? These are actually negative. And you might miss them if you're using a logarithmic scale and you don't recognize that they're slammed up against here. So this, this blue population is truly partial dim positive for CD20. All right, so, but in any case, they're both B lineage cells. And going down, once we figured out that they're B lineage, we're going to go down and take a look at our kappa and our lambda. And the red here is clearly restricted to kappa expression. So we got a monotypic B cell population, and they go from small to large, right? All right, so we can use our algorithm for that. The blue cell population is kind of interesting because it's definitely B lineage. And here, if this is my diagonal going up, I can't tell really necessarily whether this is shifted for real or it's along the diagonal. My suspicion is that it's truly shifted, and this is dim expression of kappa. So I think I've got two monotypic populations of B cells here showing two slightly different immunophenotypic features. Now, I just want to mention, let's see, once we go to the 5 and 10 on our algorithm, red is clearly partial positive for CD5. Right? The blue also looks like it's probably partial positive for CD5. For CD10, the red's clearly negative. Our blue actually looks like it might be partial dim positive. But I can tell you with certainty, because I have the control tube, that the blue is negative for CD10. This move on CD10 is due to autofluorescence. If you look, the blue, you know, they're relatively large cells, so you tend to get more autofluorescence with large cells. So these are both negative for CD10. All right, so looking at the immunophenotype for the red, I would be suspicious, could this be mantle cell lymphoma? Because if I look at it, you know, it's partial positive for 5. It's bright positive for 20, bright positive for kappa, and then it's got FMC7 and lacks CD23. So that looks like it could be a mantle cell lymphoma. The thing is, most mantle cell lymphomas are nicely positive for CD5. That doesn't mean you can't get partial positivity in mantle cell lymphoma. It's just something that catches your eye. All right, and then the blue population here shows a different immunophenotype in the fact that it's either negative or dim positive for kappa. All right, and then it looks like it's probably negative, or no, it's not probably. It's negative for 23. It looks like it's probably negative for FMC7. And I can tell you from the control that it is truly negative for FMC7. So we're dealing with, something here. We know we got a B-lineage neoplasm. We know that mantle cell lymphoma is on our differential diagnosis because it's CD5 positive. But if you remember from our algorithm, one of the other things that will often show CD5 expression is lymphoplasmacytic lymphoma. And the way that lymphoplasmacytic lymphoma typically expresses CD5 is in a dim to partial dim manner. And that's exactly what we have here. We've got a partial expression of CD5. All right. So that then gets put into our differential diagnosis. And then that kind of brings us to the point, what are these blue cells? Well, I would start to think, could these potentially be plasma cells? If this is a lymphoplasmacytic lymphoma, we know that that type of lymphoma is a B-lineage non-Hodgkin lymphoma that shows plasmacytic differentiation. So it's very typical to see plasma cells in lymphoplasmacytic lymphoma as part of your lymphoma. So let's just take a look then. Let's focus on the blue population. So down here, remember, I'd mentioned that here's the diagonal, and our blue population is either negative or dim positive for kappa. Now, if we look up here, here's my CD38 versus CD20. So here's my red population. You can see they kind of like seem like there's a connection between these and the blue. See them? The bulk of these reds sit here, and then they kind of come up and merge in with the blue population. The blue population again, and here this is the CD20 with a bi-exponential plot. You can see that it's much easier to see the true negative guys. Here you can see that it's partial dim positive, and it's picking up bright CD38. This is something that plasmacytoid cells do. And then if we move over to here, we can look at CD138, which is syndakin. CD138 is relatively specific for plasma cells in the hematolymphoid world. And it's important to remember that's only in the hematolymphoid world. There are carcinomas and other types of non-hematolymphoid neoplasm that express CD38. But within the hematolymphoid world, it's really just plasma cells and a subset of immature B cells that show expression of CD38. So these look like, based on the fact we got really bright CD38 and CD138. We've got bright expression of CD38 and positive expression of CD138. So these ought to be plasma cells, really. A caveat for CD138, it's kind of a finicky marker. You gotta be careful with it. You need to stain your cells pretty quickly, because over time you'll lose expression of CD38 in the cells. So you want to get the sample stained really quickly, and it's better not to use ACD, an ACD tube or yellow top tube, if you're going to be looking at CD138. We primarily use ACD, so we no longer look at 138, but this was not from an ACD tube. All right, so let's go back to our immunoglobulin light chains. If you look here, this was surface immunoglobulin light chain. Here's your cytoplasmic immunoglobulin light chain. And you can see, if you look at how far along the axis this blue population moves on kappa here, if we're just looking at a single axis, look at how it clearly moved way over here. So here's our diagonal here. Our population shifts off the diagonal, and it's very bright for immunoglobulin light chain. That means that we have true cytoplasmic expression of immunoglobulin light chain. So if you look, you can use your surface versus your cytoplasmic to give you an idea whether or not you have true cytoplasmic immunoglobulin light chain expression. Why am I saying that? Well, if you look at the red population here, it also shifts off the axis and shows what appears to be positive staining for kappa. All right. Well, this is positive staining for kappa. All right, but if you remember, when we look at cytoplasmic markers, we're not getting rid of the surface membrane. We're just kind of permeabilizing it. So this staining is most likely due to surface immunoglobulin staining and not cytoplasmic. That's why you have nice bright staining over here, and it didn't get any brighter on the cytoplasmic tube. Whereas here, you've got either negative to dim staining for kappa, and it got real bright on the cytoplasmic tube. So we know these are truly plasmacytoid cells. So putting the 138 positivity, the 38 bright positivity, and the cytoplasmic staining together, these are in fact plasma cells. And this did represent a lymphoplasmacytic lymphoma. So we've got basically your lymphoma cells that are going to differentiate right on into your plasma cells. Okay, I think that is the last slide I have. So hopefully that was helpful. And if anyone's got any questions, I guess you can put them on the link.
Yeah. Thanks to Buddy for joining today's session for his 20-minute talk, Buddy. but you could, you could see when Buddy starts talking about flow cytometry, the passion takes over. And hopefully you found his many examples useful. That was great, Buddy. A lot of different examples covering things. And one great thing about a video versus an atlas is that people can rewind and look at those points again and compare cases. This is going to be great. So thanks a lot. I am going to stop recording.